Well, it's nice to hear.
Okay, so I don't know which screen are you seeing? Are you seeing my?
Yeah, we're seeing the main PowerPoint.
Okay, great, wonderful. Okay, so hello everyone. My name is Janice. I'm a student in Dr. Saro's lab, and I am going to give a very brief taster on metabolic control analysis. So that is going to be the talk. So first I'm going to go over elasticities and then control coefficients, and then I'm going to try to tie it all together with a couple theorems. And yeah. So first we have elasticities. And well, why do elasticities matter? All living organisms use proteins to help carry out essential living processes, and enzymes are a class of proteins that catalyze reactions. So because enzymes are the driving mechanism behind many cellular pathways, we kind of need to know how to quantify the properties of enzymes to understand pathways. So this is where the elasticities come in, because elasticities are the quantitative property of enzymes. Specifically, elasticity coefficients describe the extent to which reaction rates change in response to environmental changes. So environmental changes can be stuff like metabolite concentrations in like media or pH, the temperature, etc. And elasticities demonstrate how a change in one part of the cellular pathway propagates to the rest. So put another way, we can say that it's a ratio of percentages. So if a substrate elasticity, a substrate elasticity of 1 means that if I change the substrate concentration by 3%, then the reaction rate also changes by 3%. And one problem with this ratio is that its value depends on the magnitude of del S. So when I say like we're going to change the substrate like X amount, then sometimes that's not a great idea, because a lot of these enzyme kinetic laws or rates, which is what V is right here, and S is for substrate, by the way. V is usually nonlinear, and that means that you can't always just take the slope of a line, because that slope of a line is not going to be the same everywhere along this curve. So in order to avoid this problem, we can use calculus to accurately describe this change in value. And therefore, we would take like the derivative of V and S, because the derivative, as you know, means like an infinitesimal change in S and an infinitesimal change in V. So again, we would also scale this ratio by multiplying with S and V. So this is how you would find your elasticity, this formula right here. And another thing to know, as I've alluded to before, is that elasticity values are unique to each point in the reaction rate. So usually when we talk about elasticities, it's helpful to know like the context of what you're talking about. So if you're saying like elasticity of when S is blank, or when S is at steady state. So here I want to stress that elasticity is this like line, and it's not a single constant, but a range of constants where individual values depend on the effector concentrations. So larger elasticities signify larger effects of the effector molecules on the reaction rate, and effectors that increase reaction rates will have positive elasticities. For example, like reactants will have positive elasticities, and effectors that decrease reaction rates will have negative elasticities, like products. So we can go through like different elasticities for different rate laws, and these are just a couple of examples. So here, this is the Briggs-Haldane equation, and this is the rate law for the Briggs-Haldane equation. Basically, this equation is based on this following setup, where the enzyme binds to a substrate to form a complex, which is what's it called? Like reversible. And then we assume that it irreversibly turns into a product and enzyme. So here are the assumptions of this Briggs-Haldane equation. Generally, it's a okay estimate for enzyme catalyzed reactions. And if we want to know the elasticity, as we discussed before, this is the rate law of the Briggs-Haldane equation. So what you would do is you would take dv ds, and then you would also multiply it with the s times v to scale it. And then we would get the elasticity this way. So as you can see, when the substrate concentration is at Km, so if S was at Km, then you can see that the elasticity is equal to half. And if it were to approach 0, you could see that the elasticity expression would be Km over Km, so that would cancel out to 1. So the elasticity nears 1 at low substrate concentrations and behaves like a first order kinetic. And enzyme elasticities would still be 1 in a reversible Briggs-Haldane reaction as well. And while it's interesting, Briggs-Haldane estimation is not applicable for all enzyme elasticities. So here, if you were to also estimate like the reaction rate in vivo per se, and this is kind of common. Sometimes you can say that you can assume that your reaction rate is equal to the total enzyme concentration times the enzyme catalytic constant. And so again, you could also find the elasticity for this reaction rate, and it's just the same way you would do as before. And so that would also give you a reaction rate, or sorry, elasticity of 1. And this is in accordance with the fact that the rate law that we started with is a first order reaction as well. So here's another example for finding elasticities for another rate law. This is the Hill equation, and the Hill equation is useful for determining the degree of cooperativity of ligand binding to enzymes or receptors. So cooperative binding refers to when the binding of a ligand to a protein is enhanced if there are already ligands present on the same protein. For example, some proteins need 2 ligands to bind to be fully functional, and having one of the ligands already bound increases the chances that the protein will be able to form a product. So the elasticity coefficient of the Hill equation can be found through differentiation and scaling. And again, you would do that and then get this answer right here. So unlike the irreversible Briggs-Haldane estimations of enzyme rates, where the low concentrations had elasticity values of 1, the elasticity of the Hill equation enzymes are equal to the Hill coefficient at low substrate concentrations. And so what you would see is that the enzymes that follow Hill equations have higher elasticity coefficients than those that follow Briggs-Haldane. And other forms of cooperative binding, like allosteric control, also demonstrate high elasticity values. All right, so you can approximate local rate law changes with elasticity values. In practice, we can approximate fractional changes when substrate concentrations are changed. So rearranging the elasticity equation, we can get an equation to make this look like an approximation, as in here. But for example, let's say that this step system is part of a larger pathway, and that outside of this pathway, there's a disturbance, like a change in upstream enzyme expression. So if this causes changes in effectors by small amounts, like del S and del P and del I, and a change in rate in del V, then, and if we know the elasticities, we can approximate like by how much the entire rate changes, provided that these del values are very small. And we can also generalize this approximation through this equation. And elasticities using this equation must be measured at the concentration that the effectors are currently in. So using elasticity values measured in one concentration in an equation set for a different concentration will yield inaccurate conclusions. Generally, for a string of reactions within a larger pathway acted on by like M effectors, so that's like just the number of effectors, the change in rate is given by this relation, provided that all effector changes are small. And as long as these changes are made in the linear regime, then the estimation works. So basically, what I want you to take home is that elasticity coefficients describe the extent to which reactions change when the environment of the reaction is changed. And the elasticity can be estimated by taking partial differentiation of the rate law with respect to the effector in question. And if you know the elasticity and the amount of change, then we can approximate the change in the rate caused by the change in the effector. All right, so now we're going to move on to control coefficients. So what are control coefficients? Well, I guess the guiding question is like, how does the concentration of one protein metabolite, or sorry, protein or metabolite influence the system's steady state? And so we want to know that, and we have control coefficients which measure the control a reaction step has over the steady state flux or a species concentration. So here, this is just a graph showing how flux changes over time as these enzymes are perturbed in, I believe this is like a 5-step pathway. And first of all, I just want to make sure that everyone knows that flux is the rate of flow of materials throughout the pathway, and it has the units of amount per time. So for example, like milligrams per second, and flux through each reaction in a linear pathway is all the same. So if you think about it, a linear pathway is kind of like a pipeline with just like segments in it. And since there's like an entire, they're just connected through like one pipe, the flow has to be the same from one end to the next. The flux through each reaction is cyclic, or branch pathway can be different. So we can't make the same assumption that we did for a linear pathway. But here, I guess the most important thing is when you perturb these enzymes, and these are perturbed separately, they will change, but then they'll land back in a new steady state. So the ending, I guess the ending is different from where they started off. So flux control coefficients answers the question. They answer the question of how much is the flux of the pathway affected by a specific enzyme. So mathematically, we express the flux control coefficients as a ratio of change in the amount of flux to the change in the enzyme concentration. And since the enzyme kinetic rates are nonlinear, the changes we make to enzyme concentrations have to be small. Infinitesimally small. So we have to take the derivative and scale this quantity just like we did with elasticities. So that is shown here. And we can also say the same for the control coefficients. These are just the ratio of fractional changes in species concentrations for a particular reaction to the fractional change in enzyme concentration in the reaction. That's also shown here, and it's very similar to the flux control coefficients. So this is the we're going to talk about control distribution, and yeah, so when all enzyme concentrations are increased by the same fraction, the flux increases by the same fraction, but the steady state metallic concentrations remain unchanged. So I'm going to prove that to you in the next few slides. So my example will be this 2-step pathway here. And in this 2-step linear pathway, the boundary species are X0 and X1. And those are fixed. Those concentrations don't change. And we know that, therefore, the flux is equal. And we also know that the flux is equal to the reaction rates V1 and V2 because it's a linear pathway. So here, when we plot the two reaction rates, V1 and V2, this is V1 and V2, not that they're, added together, but that both of them are, both of their values are reflected on this axis. When we plot V1 and V2 as a function of substrate concentration, you can see that where they intersect kind of marks the steady state point. And at the steady state point, of course, both of them are at the same rate value. And when you drop a vertical line from this point here, that kind of gives you the steady state concentration of S. So here, if you were to change the rate of either V1 or V2 by 30%, so this is changing it up, or I guess they're changed positively. So changed up by 30%, or V2 is changed up by 30%. V1 and V2 would still be equal at steady state, while the substrate concentration would change. So you can see here that since they're still like, they would still intersect, that means that their steady state concentrations would be the same, that the rates would still be the same. V1 would still be equal to V2, except that you can see that the S values, they shift horizontally, so they're not the same. However, if you did change both V1 and V2 by the same amount, then the substrate concentration goes vertically up. So that means that if you change all of your enzyme reactions by the same amount, then your steady state substrate concentration will also not change. All right. And this observation is true independent of pathway complexity. Even though I gave you an example for linear pathway, it kind of works for all pathways. So if we rearrange the control coefficients, we can also compute the changes in flux or concentration given the change in enzyme concentration. And this is just, I guess, just rearranged of the flux control coefficients and the concentration control coefficients. They're just like multiplied to each side. But again, this only holds true if the changes in the enzyme concentrations are infinitesimal. So if we make changes to multiple enzymatic steps, then the overall change to the fluxes or the concentrations will be the sum of the individual changes. Another property of control coefficients is that all flux control coefficients in a system, regardless of complexity, will sum to 1. And all concentration coefficients sum to 0. So there's an implicit assumption here where the rate of the reactions are directly proportional to the concentration of enzyme, and that the enzyme concentrations in a pathway can be adjusted independently of each other. And we can't always make that assumption, but it's still a workable assumption. So when we interpret these summation theories, they kind of imply that for linear pathways, if one enzyme step has a high control coefficient, then the rest of the steps must have smaller control coefficients, just because they do have to add up to 1. And for branch pathways, if one enzyme step has a high control coefficient, then the other steps will be small, or they could have negative. They could have negative ones. So yeah, so most reactions in a pathway will have small control coefficients, and only 1 or 2 steps will have significant control. But there's no one rate limiting step, because control is a shared property. There's like a finite amount of control available in the pathway, and it's distributed not evenly, but it's distributed amongst each enzyme according to each enzyme's control coefficient, or like their kinetics. So knowing only one enzyme control coefficient isn't helpful if you want to know which what's it called, like reaction to change in a reaction in a pathway. You can't just say like, oh, if the flux control coefficient is 5 or greater, then that's the biggest one. It doesn't work like that. If they're all 5, then the one that's like 5.1 is the one that has more control. So we can connect control coefficients to elasticities with a few relationships. These are just connectivity theorems for relating the fluxes and concentrations to the elasticities. There are 2 concentration connectivity theorems here and here. One of them describes the effect of perturbations on a common species, and the other describes the effect on a distant metabolite. And so what we know from what we can garner from these connectivity theorems is that these theorems link local effects to global effects. For example, in this two-step linear pathway, the flux connectivity can be written in the following form, like this. And the ratio of the two adjacent flux control coefficients is inversely proportional to the ratio of corresponding elasticities. So that means that the large flux control coefficients are associated with small elasticities and vice versa. So to end this talk, I guess, all in all, basically we can quantify the effect of specific enzymes and metabolite concentrations on a biochemical pathway. We can know the local control values through elasticities, or we can also find the system control values, such as the flux control coefficients and the concentration control coefficients. And the control coefficients and elasticities can be related to each other through connectivity theorems. And the control is distributed throughout the entire pathway. So the entire set of elasticities or control coefficient values must be known to deduce where most of the control in the pathways lie. Yeah.
So have you done with the talk or so? Yeah, so maybe other people have questions, I guess. Thank you for your talk. Yeah, help us clarify many definitions regarding the modeling of metabolic networks. So I'm not sure if other people have comments or questions about Janis' talk.
Do you have any kind of example that would kind of tie it all together?
As in like what kind of, like, sorry, tie it all together as in what?
Sorry, like a real world example?
Oh, okay. So yeah, I guess you can say that this is very theoretical. I guess one real world example would be, say you want to use a microorganism and put or make some value chemical. Say you want to make alcohol with a microorganism of your choice, and you make your biochemical pathway, and it has steps from glucose, and it goes through these different reactions to make alcohol. However, you don't really know like what genes to upregulate or downregulate, just because yeah, you wouldn't really know that like off the top of your head, unless it was like a simple pathway, like linear pathway. Then it's obvious that you just have to like spike in glucose to, you know, like what's it called, get your alcohol through mass action. But you also don't know like the enzyme kinetics and such. So if you do know these elasticities, if you do know like the enzyme kinetic laws, if you estimate them, then you would be able to find the elasticities and figure out, or you could, using the elasticities, you could also figure out the flux control coefficients, although you can use one or the other. And then once you know those values, you can say, oh, I see that it's like the third reaction, that gene needs to be upregulated so that I can get the most optimum amount of alcohol. I don't know if that answered your question.
Yeah,
Okay.
thanks.
Janice, I'm curious, is the definitions you have mentioned here have been applied to your PUTIDA research recently?
So I guess the short answer is yes, because and just to clarify, my research is I'm trying to figure out the metabolic pathway of Pseudomonas putida, which is a soil bacteria, and we're trying to make a specific chemical, produce a certain chemical using glucose from Pseudomonas putida. So it's like a biomanufacturing, and we don't really know the enzyme kinetics for every single enzyme in that pathway. First of all, we don't like, it would be hard to measure all the enzyme kinetics of every reaction in that whole genome. And even if we pared it down to like the core metabolism from the glucose all the way to the chemical that we want, which is called 4-ACA, by the way, from glucose to 4-ACA, that's still 80 reactions that we have to know. So what we do, I guess in the way that my work relates to this is that we do know the steady state values, and we are changing certain reaction rates by using CRISPR to turn on or off the genes. And so by collecting these steady state metabolite concentrations, if we collect them like in a large data set, we can use statistics to kind of have intellectual guesses of the elasticity values. And from there, we can further tweak our design of the Pseudomonas putida to make more 4-ACA.
But how do you get it's not the elasticity you want though in the end, is it? It's the control coefficients.
Yes.
So how do you get those?
Well, it's kind of like the same, I guess we would get them the same way as the elasticities. But for my statistical algorithm that I'm using, I actually find elasticities first and then calculate the control coefficients from the elasticities.
Oh, I see. And then what do you do with the control coefficients after you've calculated them?
Yes, so after I get the control coefficients, I kind of look through the whole pathway and look at the control coefficients and see where is the most control in this whole metabolic pathway. And if I know which genes or reactions are the most important, then I can see whether or not we can.
What do you mean by the most important? For what?
Yeah, okay, sorry. So what I mean by the most important is like, if the control coefficients are high, like if the magnitudes are high, we know that it exerts more control on the metabolic pathway. And by control, I mean like in the definition where if I change the enzyme concentration, then the flux concentration also changes by a specific amount, because it's like elasticities and flux control coefficients, they're kind of like a ratio of like, if I change this, then this also happens. So if the elasticity is 5, then if I change my substrate concentration 5%, then the reaction rate should change. Well, sorry.
So what's this got to do with control coefficients on your on the pathway?
Sorry, can
You
you say
said you had to go for the most important steps. What do you
Right.
mean by the most important step?
So those are the reactions that if you change them, or if you like upregulate them or downregulate them, they would change the flux of 4-ACA the most.
And what's that got to do with the control coefficients?
So if the control coefficient magnitudes are high,
Yeah.
then if the control coefficients of, if the magnitude of the control coefficients of the reactions of certain reactions are high, then you
Yeah.
know that those reactions are the ones that affect the amount of flux coming out of the whole pathway the most.
Oh, I see.
Yes.
So,
So
your studies try to find the steps for the biggest control coefficients.
yes, so that's the point of it. And also it's really important to actually look at them like in full before just grabbing the first 10 or like the largest 10, because sometimes the largest or the reactions with the largest control coefficient can be things that you just cannot change. Reactions that you can't change. For example, like I guess I'm just making this up right now, but if the reaction with the most control is your glucose transporter into like the whole or microorganism, you can only like the organism can only take so much glucose at a time, and you can't like upregulate that like 1,000%, like if that makes sense.
Why not?
Well, I mean, sometimes there are negative feedback loops, so like it's pointless, but sometimes
But then it'll have a small control coefficient. You
yes.
said it has a high control coefficient, the transporter, so why can't you increase the level of transporter?
Well, but there might also be other like restrictions. Like it could break it could also break other things in the pathway that you don't want, like affect growth, and then that would just kill the organism.
But that applies to any reaction step, right? Any enzyme, not just transporters.
Right. Oh, yeah, that was just like a random example.
Oh, okay.
Yeah, that's not what I found in my research. I'm just saying that that's an example.
Okay.
Any other questions or comments for Janis? So Janis, do you want to say anything more or?
No, I guess thank you for your time.
Okay, so shall we move to the second tutorial or?
It's up to you, Jen, you're the chair.
Oh, okay.
Do we
So,
want to have two different recordings or just one big one?
so I'll just stop the recording of this one probably. And then